Hello, thanks for watching this talk. I'm Eunice and I'll be presenting our research on phasmid behaviours and how they interact with morphological adaptations. Predator-prey relationships exert strong selection pressure on animal morphology and behaviour. These animal behaviours interact with their morphology to reduce detection risk. While prey motion can provide a locational cue for predators, motion can also serve as concealing behaviour in a dynamic environment. This prevents detection by potential predators or prey. Here we have the jumping spider Portia, which looks distinctly different from other jumpers. To avoid detection, it waves its palps and legs when it moves. Phasmids are conventionally known to resemble parts of plants, leaves and sticks for instance. This resemblance may vary across life stages and species, and a broad range of colourful phenotypes exists, with some 3,000 species found globally and more than 600 species in Southeast Asia. However, little is known of their behaviour. Here are some examples of the species found in Southeast Asia. They range from cigar-like species to others with beautiful blues and purples. To examine the interactions between behaviour, life stage and phenotype, we chose two species of phasmids that live in forest habitats in Southeast Asia. Loncodis brevipies is a cryptically coloured phasmid with a typical stick-like morphology across all life stages. Calvisia flavopennis is a brightly coloured phasmid that is fully winged at adulthood and are capable of flight. As you can see, both species look distinctly different at various life stages of first instar nymph, late instar nymph, and as adult males and females. We performed trials during daylight hours in this setup. This animation shows a trial. We introduce the phasmid onto a wooden dowel and allow it to acclimatize. We then bring a portable fan to the phasmid and, depending on the treatment, subject it to wind by turning on the fan. We then scored the behaviour of the phasmid. This is a non-metric multi-dimensional scaling plot used to visualize the differences in behavioural responses. Symbols of different shapes represent different life stages, with open symbols representing individuals in the control treatment and closed symbols representing individuals in the wind treatment. The orange-coloured symbols represent individuals of Calvisia, while the turquoise colour represent Loncodis. The 8 arrows in the plot indicate 8 behaviours that significantly contributed to data separation, with arrow length indicating relative contribution. The responses of Calvisia are towards the left half of the plot, while the responses of Loncodis are towards the right of the plot. These radar plots display the proportion of time that individuals spent on the 8 behaviours as seen in the NMDS plot. Only the observations of the wind treatment are displayed, as the behavioural responses are largely similar across both treatments. Mean values are given in orange, while grey lines correspond to individual observations. Likewise, the plots for Loncodis are in turquoise and grey. We found differences in the proportion of total duration of the behaviours. For both Loncodis and Calvisia, the first and late instar nymphs tend to display a variety of behaviours to varying extents in response to the wind stimulus. However, for the adults of both species, the responses are mostly confined to selected behaviours. This heat map shows the pairwise correlations between the proportions of time spent on these 8 behaviours for all individuals in a treatment. The colours and shades of the cells indicate the direction and strength of Spearman correlations. Blue shades represent negative correlation, while red shades represent a positive correlation. We group the behaviours according to their potential to conceal or reveal the phasmids. Explore, stand, and walk were classified as revealing behaviours, while extend, flatten, hang, sway, and raise were classified as concealing behaviours. Revealing behaviours are positively correlated to other revealing behaviours, while concealing behaviours are negatively correlated to other concealing behaviours. This is with the exception of swaying, which is positively correlated to revealing behaviours like stand and explore. Our explanation for these results is that once phasmids adopt a concealing behaviour, a change of behaviour could break their concealment. However, if phasmids already adopted a revealing behaviour, there could be less costs to switching to a different revealing behaviour. In fact, they could then benefit from a combination of different behaviours. For instance, a combination of exploration and walking could lead them to a better suited spot for resting or feeding. Swaying at intervals could thus provide phasmids some form of concealment between revealing behaviours through motion masquerade in a dynamic environment. In conclusion, our study provides a first insight into the variability of behavioral responses of different phasmid species and life stages to changes in the environment. Further behavioral experiments will be required to investigate the effect and function of more behaviours. Through the interaction of behaviour and morphology, phasmids may be able to adjust their adaptive resemblance strategies to specific situations. Investigating the behavioral components of predator avoidance strategies is a further step towards a more complete understanding of the complex interactions between animal morphology, behaviour, sensory ecology, and environmental factors. With that, I end our presentation. I thank you for your attention and our funding agencies and collaborators for making this work possible.